So far, we've talked about genetics in a lot of different organisms: peas, snapdragons, Labrador retrievers. We're gonna in chapter 15 talk about fruit fly genetics. But in all honest truth, do you really care about the genetics of the fruit fly that's buzzing around your fruit bowl? Probably not, right? We are mostly interested in human genetics. Why is it that I am the way that I am? Now, everything that we've talked about so far, for the most part, applies to humans. We show Mendelian inheritance patterns, right? We show polygenic inheritance. But the trouble is, when you're studying genetics in humans, humans are just not good subjects for genetic research. Our generation time is too long, right? Many couples wait for, 2-3 decades before they have kids. And so I don't want to wait until somebody is in their 30s to start my research. Parents also produce relatively few offspring, right? Families these days, you don't see families of 10, 12, 16 kids. And then lastly, my favorite is that breeding experiments in humans are generally unacceptable, right? There's a lot of ethics here. You're not going to pull two people off the street and say, well, you guys are going to have offspring because I want to write my thesis within the next, 12 months. So how do we study genetics in humans? Because again, all the things that we've talked about, they can apply to humans just as well as any other organism. So instead of looking forward to new matings, when we study human genetics, we actually look backwards. We look at the matings that have already taken place, and oftentimes this leads to the creation of a family tree. And in genetics, this family tree is called a pedigree. And in this pedigree, we don't record things like birth dates and death dates and names. Instead, we're looking for, does a certain individual in this family carry a specific trait? And when we put together this tree, then we can make educated guesses about future offspring or existing individuals and what their genotype might be for that particular character trait. And so I really encourage you to learn how to read a pedigree. This is one example from your text. I believe there's another pedigree in your textbooks as well that you guys can take a look at. But usually a pedigree will come with a key. So males are going to be squares, females are circles. If a person is affected by that certain character trait, then they will have a filled square or filled circle. When you have a line connecting a male and a female, that indicates a mating. And then any lines that go down from that mating are going to be the offspring. And so down here, this pedigree is looking at a very benign character trait. This is the widow's peak. So you can see that this particular individual, she has her hairline kind of dipping into her forehead here. That is called a widow's peak. Whereas this sister here, she has a straight hairline. The widow's peak is the dominant trait. The straight hairline is going to be the recessive trait. And so you can see here we have 3 generations. We've got grandparents, parents, aunts, uncles, and then these 2 sisters. And we identify what the genotype and the phenotype is, if known, of that particular individual. And so the filled squares or circles are going to be those individuals with the widow's peak. And then you can also see the genotype of each of these individuals. Now, when we get down to the 2 sisters here, the sister with the straight hairline, she has 2 recessive alleles because that's the recessive trait, and this particular character shows complete dominance. This sister, again, because this gene shows complete dominance, we can't tell just by looking at her if she's a homozygous dominant or a heterozygote for this particular gene. So in order to identify her genotype, we could do one of 2 things. We could either sequence the gene, if we know what it is, be able to determine what her genotype is, or we could wait until she has offspring and then, seeing what the genotype phenotype of her partner is, we might be able to deduce then what her genotype is. It's kind of like a test cross almost. All right, now when we do things like pedigrees for humans, it's we're not doing a pedigree oftentimes to see, do you have connected earlobes or not connected earlobes, or a widow's peak or no widow's peak. Usually we're more interested in disorders. Is there a disorder that is genetically inherited? How am I, going to pass it on to my offspring if I have offspring? Usually people want answers to these kinds of questions. And so what we're going to do next is we're going to look at two different kinds of inherited disorders in humans. Some are recessively inherited and some are actually dominantly inherited. And so we're going to take a look at an example of each of those. So first of all, here on this slide, we're going to look at recessively inherited disorders. A recessively inherited disorder will only show up in an individual that has two recessive alleles. So in this particular example is using the recessively inherited disorder called albinism, where an individual will have very little pigmentation in their skin, hair, the irises of their eyes, for example. So these are two sisters. You can tell one of them has normal coloration and the other one is albino. She's showing that albinism phenotype. And so in order to show that phenotype, she had to have inherited two recessive alleles, one from one parent and one from the other parent, in order to show the phenotype of the disease. The other sister, she could potentially be a homozygous dominant. She looks normal. She could carry two dominant alleles, or she could be what is called a carrier. She could be a heterozygote for this particular disease, meaning that she has one recessive allele, but she also has a dominant allele. And so because she has that one dominant allele, she doesn't show the phenotype of the disorder, but she does carry the recessive allele, which she could then pass on to her offspring as well. The other kind of group of disorders, genetically inherited disorders, that humans can have are dominantly inherited disorders. And their inheritance pattern is a little bit different from the recessively inherited disorders where you need two recessive alleles. For a dominantly inherited disorder, you just need that one dominant allele to show the phenotype of that disorder, to have the disease. Dominant alleles that cause lethal disease, means they kill you, are really, really rare and usually arise by just random mutation. But there are other less severe dominantly inherited disorders that are survivable. One example is achondroplasia. This is a form of dwarfism that's caused by a rare dominant allele. Achondroplasia is a type of dwarfism where your torso and your head are normal sized, but your limbs, so your arms and your legs, are shorter than what they should be. And so this is an individual who has achondroplasia. He's also a physician who treats patients that have achondroplasia. And so in terms of inheritance patterns, if you have parents here and the dad happens to be a dwarf, so he carries that one dominant allele, and the mom in this case is normal. If they were to have offspring, the dad, right, if he were to create his gametes, so the sperm, 50% of the sperm would have a dominant allele, 50% would have the recessive allele. And the mom, being normal, having two recessive alleles, all of her eggs would have the recessive allele. And in this case, if they were to have offspring, because the sperm are 50-50, there's a 50-50 chance that their offspring would carry that one dominant allele and would show the achondroplasia disorder. All right. And then on top of being able to do pedigrees, there are other ways in which we can test and counsel for genetic disorders in humans. So usually if a couple is thinking of starting a family and they're worried about a particular genetic disorder that runs in their family, they might seek genetic counseling. And in the genetic counseling, that might be combined with gene sequencing, for example, taking cells from the mom and the dad, sequencing their genes to see if they have anything of importance in their genes. If that's not available, but a great family tree is, then the genetic counselor could also create pedigree and do a pedigree analysis to give the potential parents some information so that they can make the decision on whether they want to start a family or not, depending on their risk of passing down any genetic disorders. Once a child is conceived, you can also do fetal testing. So that means that cells are usually taken from the womb. These are going to be fetal cells. You can perform a karyotype to look for any gross chromosomal abnormalities. You can also do gene sequencing on the fetal cells as well to pinpoint individual mutations. And then when the infant is born, there are also certain genetic screens that can be done. For all infants that are born, there's a test that's performed. It's a blood test where they take blood from the heel of the poor little baby and screams, and it's terrible for first-time parents especially. But it's a really important test because using the genetic makeup from the cells in the blood, doctors can see whether or not the infant has a metabolic disease called PKU, where they essentially can't digest a certain amino acid. And if left untreated, it could lead to mental retardation. But if it's identified in an infant, the diet can be changed. If the diet's changed, you can completely avoid any of these really terrible side effects that could occur because of the disease.